Hi, I'm Erica Ellsworth,
I'm Rob Schmidt.
And I'm Rachel Smith.
and our presentation is on the herpes simplex virus, both 1 and 2. And we'll start off with Rachel.
Hi, I'm presenting today about the basics of herpes simplex etiology. The herpes simplex virus contains double-stranded DNA, which is housed in an icosahedral capsid. It is enveloped, meaning that it has a phospholipid bilayer derived from its host cell's membrane. This applies to both types of the virus. The virus avoids detection from the body's immune system by embedding itself in the host nerve cells. While it's surrounded by the host nerve cells, it's more likely to be overlooked, ignored, or otherwise left alone by the host's immune cells. Once infected with the herpes simplex virus, it's carried inside the body for the remainder of the host's life. We'll now be discussing the cell entry. So first, a pore is created in the host cell when receptors on the host cell respond to the glycoproteins on the viroid's envelope. So glycoproteins B and C from the virus bind to a polysaccharide called heparan sulfide on the surface of the host cell. After the initial bind, the virus's glycoprotein D then binds to one or more of the host's entry receptors. The surfaces of the virus and the host cell become closer, then interactions between the virus's glycoproteins D, H, and L cause the virus to enter hemifusion with the host cell. The virus's glycoprotein B triggers the host cell's surface glycosaminoglycans to create a pore for the viral capsid. And that's how it enters the host cell. So now we'll be discussing the type 1 virions, latency and reactivation. While it's in a latent state, the type 1 of the herpes simplex virus is prone to be found in the host nerves, particularly near the trigeminal ganglia, which is a cluster of facial nerve cells. Often, while it's in these dormant clusters, the virus produces latency-associated transcript ribonucleic acid, which can be shortened to LAT-RNA. The virions expressing LAT affects the death of host cells in its proximity, allowing the viroid to manipulate the cells around it and preserve its own progeny. This effective preservation may result in recurring periodic outbreaks of a more active state of herpes. During such episodes, the virus may become transferable again. Other ways the virus can be reinstated include stress, gastrointestinal distress, exhaustion, menstruation, injury, and influenza. Now we'll be discussing the type 2 herpes and the asymptomatic state that it often enters into. Usually it is the type 2 herpes that is being referred to when the term human herpes virus is used. In contrast to the oral connotation attached to type 1, type 2 herpes is generally associated with the genital region. It should be noted, however, that the location of the symptoms displayed are not necessarily accurate indicators of which variation of the virus has been contracted. In fact, recognizing any infection with herpes is difficult, as nearly 80% of infected people do not display symptoms. This is known as being asymptomatic. Additionally, as previously mentioned, the latent state of herpes is not readily expressed through its symptoms. And that's all.
We can end it from start, right?